two identical-looking molecules, one that heals and one that harms. If you're thinking, wait, that sounds like science fiction, you're not alone. But that's exactly what happened with thalidomide, a drug once hailed as a miracle for morning sickness. But that ended up causing thousands of devastating birth defects. But what if I told you the real reason behind this disaster lies in a mind-blowing fact about the drug's molecular shape, something called chirality? And if you think you've never heard of chirality before, trust me, you're wrong. We'll get to that in just a minute. In the meantime, let me introduce myself. I'm a science journalist, and on this channel we explore the science that matters. And today we're going deep into one of the most incredible stories in all of medical history. One that led to one of the worst disasters in drug development and also helped rewrite the rules of modern medicine. So first things first, what are we talking about here? Well, when it comes to understanding the world of molecules, we often focus on their chemical makeup, what atoms they contain and how they're connected. For example, water is made of 2 hydrogen atoms and 1 oxygen atom, or H2O. Now that's a super simple example, but you get the idea. But sometimes knowing the basic ingredients doesn't tell the whole story. Picture this. Imagine your hands. They look the same, right? But try putting your left glove on your right hand, it just won't fit. That's because despite having the same components, your hands exist in different 3-dimensional shapes. They're mirror images of each other, kind of like a pair of scissors. No matter how much you twist or turn them, they just can't fit together perfectly. And guess what? Molecules can be the same way. Some molecules exist in 2 or more forms called enantiomers, which are like mirror images of each other. These are the so-called chiral molecules. And the property that makes them act this way is called chirality, derived from the Greek word for hand, kairos. So why does any of this matter? Well, because it turns out that this molecular handedness can have profound effects. In the mid-1950s, doctors were desperate for an effective treatment for morning sickness. Enter thalidomide, a synthetic drug that looked like it could be the answer to pregnant women's prayers. In clinical trials, it seemed to calm nausea and help women keep their meals down. And best of all, it appeared to be safe. But then something strange happened. Babies born to women who took thalidomide started showing severe physical abnormalities. We're talking about flipper arms and legs, stunted growth, and internal organ problems. It was a nightmare scenario, and soon thousands of affected babies across Europe and the U.S. were diagnosed with a condition known as phacomelia. The impact was devastating, and the finger of blame initially pointed towards the manufacturer. See, when the pharmaceutical company Grunenthal made larger batches of the drug, the quality control was lacking, leading to concerns that the drug had been contaminated or that different batches had varying levels of active ingredients. But here's where the story takes a fascinating twist. The issue wasn't contamination or inconsistent manufacturing. It was something far more perplexing. Delving deeper into the structure of the drug, researchers uncovered a shocking fact. The thalidomide that made it to pharmacy shelves wasn't a single form of the drug. It was a 50-50 mixture of 2 slightly different versions of the same molecule, each with its own unique 3D shape. Now, remember those mirror image molecules we talked about, the ones that are like your left and right hands? Well, the 2 forms of thalidomide were exactly that, mirror images of each other, and this discovery raised a lot of questions. Like, how did this happen? Why weren't they identified earlier? And why did only one of the 2 forms cause birth defects? To understand what went wrong, let's take a closer look at the structure of the molecule. Picture a central carbon atom attached to 4 different groups, making it a tetrahedral shape. 2 of these groups are identical, each consisting of an oxygen atom bonded to 2 hydrogen atoms. But the other 2 groups are different. One has a nitrogen atom connected to 2 hydrogens, and the other has a 5-membered ring with a couple of extra hydrogens thrown in. This setup creates 2 possible arrangements, like trying to cram 2 different shaped gloves onto the same hand. And if you look closely, you'll notice that one arrangement looks like the other, just flipped. And that means the 2 forms of the molecule, the so-called R and S enantiomers, are chiral. Each is a non-superimposable mirror image of the other. Okay, so we have 2 forms of thalidomide, but how does that explain the health risks? Here's where things get really interesting. The reason why both forms of thalidomide were present in the drug, you see, the manufacturing process inadvertently created a mix of the two during the synthesis, which is basically chemistry speak for creating the drug. And by the time it was purified and packaged, it had become a racemic mixture containing equal parts of each enantiomer. And that seemingly innocent mixture had disastrous consequences. By the way, chemists usually refer to this as a racemate or a racemic mixture. But no worries, we won't confuse you with too many big words. So the real question is, how do these two seemingly identical molecules cause such different effects? Well, here's the thing. Inside our bodies, proteins act as molecular matchmakers, bringing together the right molecules at the right places to facilitate reactions. But these proteins aren't just any old shambled bunch. They have specific shapes with grooves and pockets designed to accommodate particular molecules, and that's where the trouble starts. The two enantiomers of thalidomide fit into some of these protein receptors in similar ways, but not completely. One of the forms binds noisily and triggers the intended response. The other, well, not so much. It causes the protein to misfold or change shape. And this disruption sets off a chain reaction affecting other molecules and functions. For example, one form of thalidomide binds to an enzyme called somatostatin, which is involved in regulating various bodily processes. When it binds, it stops the enzyme from breaking down another compound called cyclic adenosine monophosphate, or CAMP. And CAMP plays a vital role in signal transduction, essentially relaying messages within cells. By blocking the breakdown of CAMP, the beneficial form of thalidomide helps reduce morning sickness symptoms. But the troublesome right-handed form doesn't do this. It binds to somatostatin, but instead of blocking the enzyme, it activates it. And here's the kicker, it does so much more strongly. So when this happens, it shuts down the usual signaling process, disrupting normal cellular communication and throwing the delicate balance of the body off kilter. What's worse, this interference isn't limited to a single process. The harmful form of thalidomide messes with other enzymes too, triggering a cascade of unfortunate events. And you guessed it, this molecular mayhem disrupts critical functions, particularly in developing limbs, explaining the tragic birth defects seen in babies born to mothers who took the drug. You might be wondering if the bad effects were tied to the right-handed form of thalidomide. Why not just isolate and sell the good version? Well, that sounds simple enough, but it's not so straightforward. And that's due to the weird quirks of molecular behavior. Even if you started with a batch of pure left-handed thalidomide, the problem is, inside the body, these molecules can flip back and forth between left and right forms, a process called interconversion. And that means if you took only the safe isomer, it could turn into the toxic one once inside the body. So here's the takeaway. A drug's effectiveness and safety aren't just about what it's made of, but also how it behaves in the body. And that includes the often overlooked factor of molecular shape. After the thalidomide disaster, the scientific community was shaken to its core. This tragedy prompted a complete overhaul in the way drugs are tested and approved. Suddenly, it wasn't enough to know what atoms a drug contained. Scientists had to understand its 3D structure and how each form could behave differently in the body. This realization led to strict testing for chirality in pharmaceuticals, revolutionizing how new medicines are developed and regulated. In the wake of this tragedy, scientists became more aware of the importance of studying the individual isomers of a drug, trying to understand the distinct effects of each. This shift marked a significant leap forward in drug development and safety protocols. The story of thalidomide is more than just a historical footnote. It's a constant reminder of the need for precision in medicine and the far-reaching impact of a single discovery. So now you know why the story of thalidomide is more than just a tale of morning sickness relief. It's a story of two almost identical molecules with drastically different effects. It's a testament to the incredible complexity of our bodies and the challenges of bringing new drugs to market. It also highlights the incredible power of scientific curiosity and the steps we've taken to ensure such a tragedy never repeats itself. And it's a reminder that in the world of medicine, paying attention to every detail, no matter how small, can mean the difference between healing and harm. As always, stay curious. and thanks for joining me on this journey through the amazing world of science. If you liked this episode, please don't forget to like and subscribe. And if you have a comment or a thought you'd like to share, leave it below. I can't wait to see you in the next one.